morning, everyone. It's my great pleasure to introduce our speaker today, Dr. Rakez. Dr. Kayed is a professor in the Department of Neurology and Department of Neuroscience, Cell Biology and Anatomy at University of Texas Medical Branch. Dr. Kayed did his undergrad study in chemistry at University of Jordan, followed by a Ph.D. in organic medicinal chemistry at University of Tübingen, Germany. He then did his postdoc research in Alzheimer's disease and protein misfolding disease at UC Irvine. Dr. Kayed's research is mainly focused on mechanisms of protein misfolding, aggregation, and toxicity in Alzheimer's disease and other neurodegenerative diseases such as Parkinson's. And his lab has made the seminal discovery of tau oligomers and has since developed therapeutic approaches to target amyloid and tau oligomers. And Dr. Kayed has published over 150 papers and has won numerous awards, including the very prestigious Inge Granke-Ickable Award for Alzheimer's research this year at AAIC. So we're so glad to have Dr. Kayed speak with us today. And the title of his talk is Brain-Derived Biological Relevant Tau Aggregates in Alzheimer's and Tauopathies. Let's welcome Dr. Kayed.
Thank you, Dr. Chen. That's a nice introduction. It's always a pleasure to talk to people at UC San Diego. I know a lot of you guys and the great work you all doing. So I chose to talk to you today about brain-derived biologically relevant tau oligomers because, you know, now there is structures of the fibrils derived from Alzheimer's brain and different tauopathies. So, and I want to try to convince you that we should do the same for the non-fibrillar materials. So, as a disclosure, I'm an inventor on patents involving tau oligomers and antibodies that are specific for tau oligomers. So this is kind of an outline for the talk. I think everybody now agrees that protein aggregates in disease are heterogeneous. And I don't want to call them strains because, you know, it may imply infectivity. So we call them polymorphs. And these polymorphs, normally they are dynamic. They can change during the course of the disease or under different conditions. And then I'm going to show you that you can isolate actually the non-fibrillar aggregates too, not just the fibrils. And I want just to touch on what's the cause of the polymorphs. And one thing we are trying to look at is if there are different amyloids, they may interact and this may result in a different structure. And perhaps we'll touch on the implications for diagnostics and drug development. So I think I don't need to spend a lot of time on these first few slides, especially the pathology. We know the hallmarks of Alzheimer's disease, for example, there's the neurofibrillary tangles and the amyloid plaques. And if you zoom in the neurofibrillary tangles, they are normally not all classical. There are different shapes. There are the Brie tangles, the grains, the neurofill thread, etc. So even at the microscopic level, we can see that these aggregates are a little bit different. And if you go further, you will see that in the human brains, there is different tau aggregates. Does this microscopic or the images reflect differences at the atomic level, how the protein aggregates or the fibrils form? So that's the big question. And the simple answer is yes. So these microscopic differences reflect different orientation of the tau aggregates. And as we know for tau, there's the 3R, the 4R. So it's also a big debate. Is it one or the other? I think based on some in vitro data and recent structural data, that these two can cross-seed and both aggregate. And as we know, there are like BSP, there's 3R, and in Biggs disease, there's 4R, but also they coexist in different diseases. And it looks like they aggregate both of them. So, as I said, first started to look at the microscopic level, then Mark Diamond work. When they isolated from different diseases, they show that they are different. They have different seeding propensity. They can cause different pathologies. And all these studies suggest they are different and they have different biological activities, most likely. And then the big breakthrough came when the Goddard group and his colleagues, they solved the core structure. And I want to emphasize, this is the core structure of tau fibrils isolated from Alzheimer's disease. And this is a huge discovery because now we're not speculating or just using low level biophysics. These are atomic structures. And they followed this breakthrough and they took advantage of the advances in cryo-EM technology. They solved the structures of so many proteins, so many protein aggregates derived from human brain, also aggregated in vitro under different conditions. So now we can see this is the pair helical filament. You can see also the single filament, how they come together. And they solved it not from Alzheimer's disease, but also from CTE, from Biggs, from CBD, and so on. So now if you have tau fibrils prepared in vitro or isolated from diseased brain, you can, it's a hard work, but you still can solve the atomic structure of the core of these fibrils using cryo-EM. And recently, because then, okay, sometimes for some experiments, you cannot just isolate material from brain, you may need to propagate it. And the knock was always, if you propagate material in vitro, it may not resemble the ones in the human brain. But we know now that at least the fibrils are faithful in their propagation. Like if you take specific fibril from a human brain under the right condition, you see the recombinant protein, you can get similar atomic structure. Having said that, we have to be cautious because there may be, they are missing the post-translation modifications. But at least under these conditions, which were beautifully described by two groups, one for tau, one for A beta also, you can reproduce these brain-derived material in vitro. And they went further even to try to subgroup or classify tauopathies based on the core structure, the fibrillar structure. One other thing I want to touch on, and it will come, we will talk about it maybe later, that at least in cell culture, Todd Goldie's group showed that these tau aggregates in cells, they are dynamic. So, and this was shown for A beta by multiple groups in vitro and in vivo. So if you have an amyloid, there could be an equilibrium or there could be dynamic transition from one conformation, one structure to the other. So this is still under investigated, I would say, for tau, but it's important to keep in mind that you can manipulate these aggregates in vitro, but also, at least for tau now in cells, they can change. For A beta, it's shown by 2-photon microscopy in mouse model that there is dynamic, that the blacks may break up a little bit, or they may recruit, or they may disappear. So we have to keep this in mind that amyloid structures are dynamic. Now that's about the fibrils. What my lab and Skip Binder also looked at, like almost now 12 years, that there is what we call tau oligomers. And to define these, Oligomers, as you all know, it's a vague term, but we try our best to put definition. So at the lower limit, there are 2 to 3 tau monomers. At the upper limit, there are 8 to 12. Regardless of their both transition modification, I'm saying this like if they are 3 to 5, if they are acetylated, phosphorylated, still they are oligomers. But this does not mean that the phosphorylation or acetylation or ubiquitination doesn't affect the formation of these aggregates. But when they are in this size and the morphology is non-fibrillar, which means they are still in the spherical or protofibrillar structure, then we call them oligomers. What we know about these oligomers that their conformation is distinct from the fibers. The fibrils, most of the time, they anneal, they are bare helical filament. These are just spherical or rectangular aggregates. They mediate pathology spreading and cytotoxicity. They display hydrophobic surface patches. They're flexible and dynamic. So these are the generic terms for them. Now, spreading and cytotoxicity, they are mediator of this compared to the same amount of unsonicated fibrils, which I'm going to show you. Now, if you take the fibrils and you sonicate them mechanically, then you produce these small aggregates, including oligomers, then for sure you will see spreading and cytotoxicity. But if you leave the fibrils in their native bare helical large structure, they don't. They can be detected in tissue, biological fluids, isolated from human and mouse tissues. They are mainly enriched in the BPS fraction. The drawback about oligomer, they are hard to quantify, and there's still no atomic structure like the ones I showed you snips of for the fibrils. Now, the term, we have to give credit, it came originally from Takashima group. They were the first to show these oligomers. And after that, my group here and Skip Binder, we both characterized the same species in Alzheimer's disease brains. And it was almost exactly the same, these dimer trimers, and there's the sexamer. And they were enriched in AD compared to control. So this is the first description from my group and Skip Bender about them. We developed antibodies and he developed reagents and assays too. So this is where we think the oligomers started, or tau oligomers. And now there's so many groups looking at them, and they discover them in human brains and mouse brains. As I mentioned here, they're still hard to quantify and no atomic structure. So if you want to work with oligomers, you better be patient. And hopefully at the end, I'll show you that there will be some resources to help you or anyone who wants to join this exciting field. Now, as I mentioned about mediator pathology spreading and toxicity. So Christian Lasagna-Reeves, who has now his own lab in Indiana, was my first graduate student. And when Christian and other lab members took a human brain and they isolated these oligomers, and they isolated the fibrils from the same Alzheimer brain, frontal cortex, and then they did a lot of experiments in vitro, but even when they injected them in wild type mouse, these oligomers mediated the spreading of the pathology. They injected in the hippocampus and then the pathology spread all over. These only caused some local spreading, but there was no pathology in distal spreading. So only local spreading. Now, I want to emphasize that these were unsonicated, but if you sonicate the fibers, definitely you will get spreading and pathology. So it's not one or the other. It's just, you have just to control that the fibers are unsonicated into smaller pieces. And so the search for these non-fibrillar aggregates did not start with our work. It was already there before. People were looking why some mouse model have behavioral deficit before the NFTs form, etc. And this is almost all the mouse models. Also, synaptic dysfunction was shown before or independent of the neurofibrillary tangles. And now, as I said, you can look at these 88 positive phosphorylated neurofibrillary tangles, and these are the tau oligomers. These are in human brains. So they can be detected, but also they are distinct. So the oligomers normally, they are punctate diffused aggregates, while NFTs and other fibrillar aggregates, they are more compact and easier to detect. Now, the other thing which emphasized about the spreading, this work from Karen Duffy's lab when she was in New York, and she showed that only the small aggregate, so size is important here. Unsonicated fibers did not do it, but sonicated fiber or small chunks of oligomers, they can be axonally transport, and we think this is important for the distal spreading of tau pathology. Also, a lot of work, this is from Merck, they also show the same thing, these dimer-trimer species in stem cells, so they are, so there is a lot of evidence about the role of these non-fibrillar aggregates in tau pathology, and hopefully more discoveries will be made about the exact mechanisms. And this is just a lot of emphasis on it. Now, as I said, it did not start here. Actually, the Iqbals, long time ago in 1993, they tried to find this non-bare helical filament pool in Alzheimer's disease. So it's really good to find these papers where they used elegant biochemistry to try to understand what's the role of non-bare helical tau in Alzheimer's disease. Now, how we isolate these? Okay, we published and many groups published that you can prepare tau oligomers in vitro. And similar to fibrils, you can use heparin. For oligomers, you use different mild conditions and you stop the reaction and you get recombinant oligomers. But the challenge was to isolate these oligomers from human brain. So we followed two methods. One of them is reagent dependent. We do immunoprecipitation. And the other, we use just sucrose fractionation. This is just standard sucrose fractionation, and you will find these oligomers enriched in fraction 4 and 5. So these are the two methods. So for immunoprecipitation, you use an oligomer-specific antibody, you will elute them, and then you get these purified oligomers, you characterize them, and you use them to do your experiments. And for the sucrose, the same, you can purify them and they're pretty pure. So these are the two methods to isolate them. And not just our lab, Brad Hyman isolate these oligomers using sucrose fraction. They call them high molecular weight aggregates. So, but eventually they are the same range of species. Once you isolate them, you can purify them, and then you can do a lot of characterization to try to see if you isolate, for example, from Alzheimer's disease versus PSP versus DLB, are they the same or different? One thing about these methods, it looks like the aggregated tau formed in the human brain is more stable. So we did a lot of controls. They don't change much, the brain derived. Like they're stable for almost 1 week at 4 degrees. If you store them in -80 or -20, you can keep them up to 6 months. But if you keep them for longer, they will convert to different aggregates, especially larger aggregates, which become less toxic. So the methods for isolation and characterization, we published 2 book chapters about them, and there are detailed protocols. If somebody has any question, he or she are free to email us. We will be happy to help you to isolate them. And as I mentioned, we isolated them first from Alzheimer's disease, then we expanded to BSP, and then dementia with Lewy body. And this was important because we wanted to see if the oligomers, similar to what was published for the fibrates, they are different in these different diseases. And we followed first standard biochemistry. So basically, once you isolate from Alzheimer, DLB, PSP, this is A beta oligomers as a control. The first step is to do a panel of antibody. You can use conformation antibodies like the ones we develop, and also you can use commercial antibodies like something against N-terminal versus the middle region versus the C-terminal. So there's a lot of tau antibodies, but I don't think you can depend on one single antibody. You have to look at different ones. Then we performed also atomic force microscopy to look at the size or the average size. And you can see clearly from the analysis of large number of these single oligomers, they are different. Size exclusion chromatography, they are almost the same size here, but there are some other differences you can detect here, especially in the AD. You can perform circular dichroism. This just gives you how much beta sheet or random coil or structure in the oligomers. Remember, the oligomers, they have flexible region. They are not all like ordered beta sheet structures. FTIR also makes differences. So all these tells us that initially there could be different polymorphs. And one other thing, and this is recent data in collaboration with Vicky Yunchinsky at Ohio State, and we can see that despite what I said, they are heterogeneous. If you do size exclusion chromatography and you purify them, you get pretty homogeneous peaks. And this is important because for structural studies, solid state NMR or cryo, you need high concentration. So this is why we think they are homogeneous, at least at these low concentration preparations. Okay, that was just like, I would call it superficial evidence that there are different, but then you can follow what the Brion people do. You can do digestion and see if the core which is resistant to digestion is different. And this is normally the standard assay to say these two are different strains, and I said I want to call them different polymorphs. So you can digest in pronase, and here the way you look at these are in two different things. First, you have to do first time course, so under the same concentration of the pronase, and see which one disappear first. So the ones which are more resistant will stay here. The ones which are less resistant, they will disappear into smaller fragments. So this is the first thing. This tells us about the resistance, but doesn't tell us that the core is different orientations. This, you have to look here if they have the same fragmentation pattern. So these are the standard assays. And it's clear from these, the pronase digestion and the proteinase K digestion, that these are different polymorphs. So the oligomers for different diseases have different fragment patterns, and they have different resistance to the enzymes. So now we are almost sure, without the atomic structure, that they are different. They are polymorphs. In order to confirm it, we followed a recently published protocol from Claudio Condello at UC San Francisco. And this is the fluorescence amyloid multi-emission spectra. And the beauty of this method is that they use reagents which are available, like you can buy them from anywhere. Thioflavin T, we all know about it, curcumin, FSB, and we added to his protocol BCNS because we are interested in the hydrophobicity. And the first experiment we did is to try to compare the brain-derived oligomers with the recombinant tau oligomers. So we took 4R tau here and we aggregated into oligomers and we isolated oligomers from Alzheimer's brain. And you can see the clear difference between them. So, and if you want to stretch it a little bit, you can see this is, the analogy is the heparin fibrils versus Alzheimer brain-derived fibrils, and they were different. So at least we can see also the oligomers from recombinant versus Alzheimer brain, they are different. And you can do a lot of the principal component analysis, etc., and confirm it. So that was the starting point. Then we compared the oligomers isolated from different diseases and recombinant 4R oligomers, and you can see there is overlap between them. So the separation is not as clean as it was in Alzheimer brain-derived oligomers and recombinant oligomers. And this also, you can use the same method to look in tissues. So it's exactly the same method, and you can look at them, and they are different reactivity with the different compounds. Okay, so this is basically all the evidence we have so far. We are working hard with Lemhan at Eastern Carolina to get the cryoatomic structure for it, but we are making progress, but we're still not there. So what about the biological activity? So you can prepare things which are different, but if they are not biologically relevant, so what's the point? So the first step is to look at the standard, which I think everybody use, Mark Diamond biosensor cells. They overexpress only the core of the tau protein, but they are widely used. And with this experiment, you have to look at two things: the number of aggregates and the shape of the aggregate. So these are the two criteria to say that these seeds are different, because they may give you larger number or they may give you different morphologies. And you can see from the analysis that, yes, they have different seeding potentiality. And then the knock is these cells, as I said, they overexpress only the core of tau, the tau protein. So Nisha in the lab, she's talented in primary neurons. So she took primary neurons from H-tau mice, also wild-type mice, and she can see or assess the seeding potentiality in primary neuronal culture. And as you can see from the analysis, the PSP has the highest, but also, you know, it's hard in the primary neurons, unlike the other cells, to say that the conformation or the aggregate shape is different. But at least you can see which one is a more potent seed. Now, the other thing, as we all see it as important, is the neuronal activity. And for this, you can use these brain-derived oligomers and use the LTB, see how they affect the LTB in brain slices. These are well-established methods. They were studied extensively for A-beta by Dennis Selko and others. And the beauty of this, first you can do a dose dependent curve. So you can see at low concentration, nanomolar concentration, you can see that these oligomers in a dose dependent matter, they inhibit LTB. This is from Alzheimer's disease. Then you say, okay, I want to compare the different diseases, and you can take the 3 different oligomers from 3 different diseases, and you can perform it, and you can see they have different activity. Now, all these in vitro or ex vivo, then The ongoing experiments, which we had to repeat for a second time, is once you inject them in animal, do they give you pathologies similar to the disease they are derived from or not? So far, we don't have conclusive evidence that the PSP will give you PSP-like pathology, but they are biologically active in vivo. All right, so the other thing when you use brain-derived oligomers, it is important because at the end of the day, we all want to develop therapeutics or diagnostics. So you can use them to screen if you have antibodies, if you have small molecules, if you have aptamers, you can use them to screen which reagent is important against which aggregates. And as we know, at least from the BIT imaging, that the BIT reagents which worked for Alzheimer's disease did not work for BSP. So even in live vivo. So this is a crude, but I think it's an important method to screen. So for us, we developed what we call them tau oligomer monoclonal antibodies. We have 4 antibodies. One of them we published extensively. And you can use these brain-derived to see which one reacts with one. As you can see, all these antibodies don't react with A-beta oligomers. And so this is the first, is just to use simple ELISA. But then you can pre-incubate these oligomers with the antibodies and then add them to the cell and see if the antibody binding prevented these aggregates from seeding, from having effects on LTB. So this is one way to use it. And this leads us about how we can target tau oligomers by immunotherapy. We did not start this. Like, as you know, Sergeson and Hannah Rosenman and others used active and passive immunotherapy. Many groups tried to target the phosphorylated aggregates. And based on these preclinical studies, there's a lot of patents and companies. They're going into clinical trials. I think this is the latest and the best review to summarize these is in our Sergeson current recent review in drugs. And so which one targeting the N-terminal was terminated in AD, but I think still ongoing in BSP. I may be wrong on this one. So there is a lot of clinical trials trying to target tau. And basically, if you look, these are where the epitopes for these different antibodies lying. So as I said, one of the N-terminal failed, and the rationale behind this was there's a lot of N-terminal tau in the CSF, but the ones which looks like promising are the one targeting the core of the tau protein. Hopefully they will work. For us, we are interested in targeting oligomers. And we published a lot using TOMA1, which is an IgG2A antibody. And we did it in different mouse models. And the dose we needed to target oligomers compared to fibrils or all tau species was almost 1/5 to 1/10 of the doses. And you can reverse it in one single ICV or IV injection. And we repeated this. It did not work in the TG4510, so that was a negative result, but the rest were fine. And also even we reversed it in Parkinson's mouse model. Now, the knock on the tau oligomer antibodies, so what do they bind? So we, with the help of collaborators, we used the random library. So, and this is, I'm showing you the epitope for the first time, I think, in my presentation for TOMA1 and TOMA3. And you can see they bind non-continuous epitopes. So all 4 clones did not show anything in the N-terminal, but this is TOMA1, so it has 3 non-continuous epitopes. And we think these 3 epitopes are displayed when they are in the oligomeric state. When they are in the fibrillous state or monomeric state, these are at least not. And this is, I have to even to mention that this is just one method. We're still trying to collaborate with somebody to do NMR and confirm these epitopes. This is for TOMA3, so there's a small epitope here, and there is the major epitope is here. Now, why we wanted to look at different antibodies? Because we think there may be different strains, and this is why, for example, when we immunized 8-month-old H-tau mice with TOMA1, we could reverse the phenotypes, all of them, at 8 months. But when we aged the animals up to 20, 22 months, we saw that a single antibody may not reverse all phenotypes. So, and this paper just came out last week, I think, or 2 weeks ago. So we think basically you need to develop many antibodies, many aptamers or small molecules if you want to target the different strains. And this will be the case until we determine which are the ones, like the major ones, which are disease relevant. Now, immunotherapy is not the only approach you can use. You can use different approaches, and there's a lot of excitement, especially if you are enhancing the protein. I think you can degrade a lot of the intracellular aggregates. Immunotherapy, at least for now, it's mainly targeting these seeds which spread from one to another. And when I say one cell, I don't mean neuron to neuron, neuron to glia, glia to neuron, etc. So there are many approaches. I still think if the strains or the polymorphs hypothesis is correct, I think passive immunotherapy most likely will be the best approach for the extracellular tau. Now, I'm gonna run through these few slides fast. So now it's confirmed that in these brain tissues, you don't find just A beta and tau, for example, in Alzheimer's disease. There is alpha-synuclein in almost 58%, recently PDB43 in more than 50% of the cases. So it looks like these are the 4 major protein, and this is from the NIA summit in 2019. At least there are 4 different proteins. I think there are more, but these are the key ones: amyloid, tau, alpha-synuclein, PDB43. And one way we wanted to think about is that perhaps based on which combination, which ratios, or how they are co-aggregating, maybe they affect the disease, and maybe also the overlap in the symptoms. But this is, I'm not clinician, so I cannot judge this. But it's clear that these diseases, there is overlap between them. And for example, the synuclein Parkinson, now we say there's synuclein Alzheimer's. Tau is mainly in Alzheimer or PSP, but now there's in Parkinson disease, there is tau aggregates. TDP43 also the same. Also A beta, if I remember correctly, was found in Alzheimer's disease, in Parkinson's disease too. So there is a lot of pathology, elegant studies showing and quantifying the different protein aggregates in different diseases. And it looks like the implication, most likely you will have to target most of them, not just one of them, at advanced stages. Maybe at early stages, as we see, there is promise for just targeting A beta and maybe A beta tau. But if this is true, at late stage, you may have to target at least a couple of them. Or you can buy antibodies, or you can use different cell mechanism to get rid of the aggregates of all these proteins. So the most studied relationship between 2 different protein aggregates is tau A beta. And this is for a long time. We know that if there is tau knockout, you add A beta, it's not toxic. So people suggested A beta is the trigger, tau is the executioner, etc. But even in recent studies, like in using PET imaging, you can see that they interact, or once they are both in the same region, you start to see neurodegeneration or effect. So, and also there is work from Tara Spire-Jones' lab showing that it's not that they are both there, that there is some synergy between them at the synapse. So the fact that both of them synergize is exciting, and it looks like you have to target them both. Now, for us, this is more, the co-aggregation is more widespread than just A beta and tau. We looked a lot on synuclein tau aggregation, and we think even this relationship is really important for mediating synuclein toxicity. Now, there are some evidence that maybe it be direct or indirect, but we see it direct, that synuclein aggregates also needs tau for mediating its toxicity, at least in vivo. There's also work from Ben Wolzen's lab showed RNA binding proteins interact with tau. And if you remove TIA, then tau oligomers form larger aggregates and become non-toxic. And this also were expanded in this Nature paper from Mike Henneke, that there is a cross seed between amyloid and the specs. So there's a lot of them. Now, what's the consequence of this? Most likely, at least based on what we know now, it leads to the formation of unique toxic aggregates. And most likely also based on our work and Ben Wolzen work, that it lessened the formation of non-toxic large aggregates. So these are the two simplistic explanations. Now, what's unknown yet, does the co-aggregation impair the clearance mechanism, or how can we target both or these mixed aggregates? So this is still unknown, the ones in red. Now, also 2-3 weeks ago in collaboration, this is from Ranjachari groups, actually they saw that TDP43 and synuclein co-aggregate, and they did elegant NMR studies and analysis, and it looks like maybe even at 1-to-1 ratio. So these co-aggregates will get more attention soon. The problem is how relevant they are in the disease process, or can you isolate these hybrid or heterotypic aggregates from human brains and understand more their role in the disease progression. Now, for us, we just wanted to look if these co-aggregation or cross-seeding is important for the formation of different tau morphologies or polymorphs. So it's a simple experiment. You take tau monomer, you can take 4R or 3R, or you can take 1-to-1 ratio 3R, 4R, and you throw at them preformed tau oligomers, A beta oligomers, synuclein oligomers, TDP43 oligomers, and then you follow the same analysis I described previously, and you try to see if these are different polymorphs. And it looks like they are. So this is just symbol BCNS to see the hydrophobic surface, and you can see there are differences depending which one you use to seed them. And as I said, we use 4R as here monomer, we use 3R, and we use 1-to-1 ratio. These are simple in vitro experiments. And then you subject them to proteinase K digestion, etc. And you can see there are different Better's fragments, not as clear as the ones from the brains, but there are differences. And then you can follow this with proteomics, and you can see, you fragment them, and you can see there are differences. The synuclein one, for example, compared to the TDP43, these are very clear differences. So on and so forth. So you can at least suggest that the co-aggregation or cross-seeding can lead to the formation of unique structures. This is not the only thing. Unique structure can form also based on the cellular environment, the host, etc. So there's multiple, and also genetics, because it looks like ApoE for tau is different than the ApoE3 tau aggregate. So there are a lot to look at. The same, as I said, we do functional assays using LTB recordings, and you can see the ones which are seeded with different seeds, they have different LTB inhibition activity. Now, this is, I think, my final 2 slides. I just want to emphasize that, yes, solving the cryo-EM of the fibrils is really, really a huge step. But we have to keep in mind that this is just the core. So you may be able to design small molecules to detect the different tau aggregates, etc. And this is, I think, where it's going, and this will be useful for bit imaging, etc. So, but one thing we should not forget is that this is not all the protein. So you have the core, but you still have the flanking regions. And most likely that the flanking region also needs to be understood, or we need to understand they are folding or misfolding in the different fibrillar aggregates or oligomeric aggregates. I think that will be key, because this flanking region is very important for the elongation, for other stuff. This is a nice review if somebody's interested from Olamic and Ital in this year. And I think we totally agree that the core is really important, because it confirms the differences of the fibrils, morphologies or atomic at the atomic level. But there's still a lot of amino acids here need to fold or misfold in the right way. So that's the last message I have. The other thing, I did not show you any of this data, but we all, when we try to study A beta oligomers or tau oligomers or synucleinomers, we focus on the neurons. But here we try to look at tau oligomers and astrocytes. And we found that actually tau oligomers, through the release of HMGB1, high mobility protein 1, can induce senescence in cultured astrocyte and in vivo. So this is really important, that when we look at the tau oligomers or non-fibrillar tau aggregates effects or role in the disease, we have also to look at non-neuronal cells, and this is what we are doing right now. I think, the last thing, as I said, if somebody need any help extracting or brain-derived oligomers or tau oligomers, recombinant, you can email me. I'll be happy to provide them, no questions asked. But also now NIH funded us for a U24. This is my lab, Jeff Corritt lab at Ohio State, David Eisenberg at UCLA, and Derek Keeney at Washington. So our job now, we are in year 2, and we hope to go live on the web soon. So our job is to isolate. So we receive from Derek Keeney like large brain pieces. We isolate them in my lab and in Jeff Corritt lab. Then we send them for David Eisenberg. We send them cross-lab conformation, then for David Eisenberg at UCLA to confirm them. And if they are what we want, the oligomers, also we are isolating fibers, but the focus is the oligomers, then we are supposed to package them and distribute them to research labs free of charge. And hopefully in a year or so, you can just go on the website and ask us for small aliquots and you will receive them. And finally, I could not have described this work without the great lab members, the current lab members. And also, I cannot forget the contribution of Ormi, Christian, Diana, the pioneers in the lab who now have their own careers, and all of them have been amazing. The support from especially NIH, and also we started with smaller foundation grants. And I thank you all for listening.